Okay, welcome everybody. Good morning for the start of this academic year's seminar series on human evolution and the Paleolithic. It's a pleasure to welcome Bence Viola here. Bence is currently based at the University of Toronto, and but some of you probably know him from a long time ago. He started up his career here in Vienna, actually, I mean, not in this building, but in the Department of Evolutionary Anthropology. At that time, I think it was had still a different name, even, and so basically the prehistory. Bence, after his PhD here in Vienna, moved for a postdoc to the Max Planck Institute for Evolutionary Anthropology in Leipzig, and then finally on to Canada, to the University of Toronto, where he's based since. Thank you very much for joining us today, Bence, and I'll give the
word. Thank you. Thank you very much for having me here. I'm especially happy to be here because I was thinking when I was preparing this talk, I was thinking when was the last time I talked in Vienna? And it was actually 16 years ago. When I defended my PhD at the time in the Altlandstrasse, and many of the people who are here now, or at least some anyway, were there as well. So it's very nice to see so many friendly faces. And then the other thing that makes me really happy is that so many of the people who worked on Denisova, who worked on some of the things I will present today, are also here. They weren't in Vienna at the time, but they moved here since then. Tom, Katharina, Susanna, Ben, Martin. Yeah, there's a lot of people. So it's lovely to be here. And I'm really looking forward to telling you a little bit about what we learned about Denisova in the now already 15 years that this site has been in the focus of our attention. So Denisova Cave is a site that I probably don't have to present to too many of you. It's in Central Asia, in the Altai Mountains, more or less on the border between Russia, China, Mongolia, and Kazakhstan. It's a large cave in the valley of the Anhui River, situated about 20 meters above the valley floor today. The site has been excavated since the 1980s. This is kind of the view of the Anhui River Valley from above the cave. You see here the scientific camp that our colleagues from Novosibirsk built there. So the Russian Academy of Sciences, their Institute of Archaeology and Ethnography in Novosibirsk, has been excavating Denisova since the early 1980s. It was first discovered by a paleontologist called Ovodov, who was looking for cave bears. And while looking for cave bear bones, he also found some stone tools. And so Anatoly Derevyanko and colleagues have been working there since then. This is the entrance of the cave. Inside the cave, you have, so this is the entrance here. You have a large central hall, the so-called main chamber, and then two areas on which most of the research in the last about 20 years focuses, the east chamber and the south gallery. The Denisova Cave, of course, is primarily known for its hominin discoveries. Again, I'm not going to spend too much time on the stratigraphy. The stratigraphy is very complex. The three different parts of the cave have rather different depositional histories. The numbering of layers is somewhat confusing because layers that are contemporaneous have very different numbers in different parts of the cave and so on. It's something you have to be careful for. From all three parts of the cave, we have some fossils that are relevant here. I'm not going to talk about the Neanderthals today, which of course are also present in Denisova, only about the Denisovans. You have Denisova 2, the very first discovery, deciduous molar that we'll see in a moment, found in 1984 in the main chamber. We have Denisova 3, the finger bone, found in the East Gallery, Denisova 8, a molar in the East Gallery, and then we have a bunch of fossils from the South Gallery. The dating of Denisova is something that has been disputed since the 1980s. This is the most recent attempt at dating by Jacobs and colleagues using a combination of OSL and other methods, also including some genetic data. As you can see, you have here the stratigraphy, kind of the very simplified stratigraphy in each area of the cave. You have time here, and then the Red, blue, and orange are occupations by different hominin groups. Wonderful thing in Denisova cave is that you have really exceptional DNA preservation, and this is why this site became so well known. In the end, especially the east chamber, one can see almost as a natural fridge. It's deep inside the cave. You have year-long 7 to 8 degrees Celsius maybe in there these days. And this is very conducive to DNA preservation. We have DNA not only from fossils. The fossils are here, the little numbers in circles. But we also have DNA from sediments, which are the triangles here. And so we know that the earliest occupations in Denisova were probably Denisovans, but soon after Neanderthals arrived as well. And then we have kind of an alternating occupation by Neanderthals and Denisovans. We also know that if not in Denisova cave, somewhere nearby they also met, because we have one individual, Denisova 11, Denny here, who is a Denisovan Neanderthal hybrid. This is an individual who had a Denisovan father and a Neanderthal mother, and is really in the fossil record the only known hybrid between two such groups. So just to kind of summarize the fossils that we'll talk about here today, we have Denisova 2, deciduous molar, Denisova 3, a phalanx, Denisova 4, 8, 25 and 29 are molars. We'll look at them in a bit more detail, and then we have some small cranial fragments as well as a radius midshaft. So the first tooth here is actually the first fossil from Denisova that I ever saw. In 2002, Anatoly Derevyanko came to Vienna. There is this very, very early connection with research in Denisova with Vienna. Horst Seidler, who was at the time the professor at the department, went to a conference on ice mummies and met Derevyanko there, who was presenting research on Scythian ice mummies from the Altai. And one of the things that came up was that we are doing interesting human evolution research in Vienna, and so a collaboration started. So in 2002, Anatoly Derevyanko came to Vienna with his wife, Tatiana Chikisheva, and they brought the material from Denisova cave and Okladnikov cave to Vienna, which then later on ended up to be a large part of my PhD. So this was the first one that we saw. This fossil found in 1984 was first mentioned by Christy Turner, an American dental anthropologist, who described it as probably first lower deciduous molar. He said that it's morphologically not very diagnostic, and he also proposed that some of the traces of wear that we see on it are similar to what we see in Arctic hunter-gatherers, strong chipping in populations that often consume bone and other hard substances. When I analyzed this, I got slightly different results. I think it's a lower second molar, again, based on very small features such as the presence of a tuberculum tucacandle here. What was interesting about this specimen is that it's very large. You can only measure its cervical diameter, but it was very large. And so, before we had genetic data on the Denisovans, I assumed it's a Neanderthal. In size, it kind of fits with Neanderthals. It's significantly larger than most recent or upper Paleolithic modern humans. In 2017, we were also able to extract DNA from this specimen. This was work led by Vivian Sloan at Max Planck that actually showed that this was a Denisovan individual as well. The most famous fossil, of course, is Denisova 3. This is the fossil that kind of started the Denisovan craze. Discovered in 2008, at the time of its discovery, it was a relatively complete phalanx that was then cut into 2 pieces. Sadly, the photos that were taken before it was cut were lost. So we have no idea how it looked completely. And then the 2 pieces were sent off to 2 different labs. The proximal end here ended up in Leipzig, so we have micro CT scans of it. The other half, first, it went on a world tour. It first went to the US, to Eddie Rubin's lab. It then went to Paris, to Eva Maria Geigel's lab. Eva Maria Geigel managed to extract mitochondrial DNA from it and actually also took good photographs before she sampled it. So these are the only photos we have of the distal part. And then it was sent to Copenhagen, where it was supposedly ground up completely without any further scanning. So we're missing the second half. Of course, it's a little hard to do morphological analyses on such a fragmentary piece. What is clear based on the micro CT scans of the proximal end that was clear to us is that this is a juvenile. It's actually the epiphysis and the diaphysis are just starting to fuse. So in modern humans, this would be an about 14, 15-year-old. What's interesting about it morphologically is that the distal end is very, very slender. In Neanderthals, phalanges usually have very wide, kind of mushroom-shaped distal ends, which you don't see in this piece at all. And so in 2019, we did a morphological analysis based mostly on photos and measurements we could take from these with Bennett and Eva Maria Geigel and colleagues from Paris. And we could show that Denisova here in this principal component analysis of I think it was 12 measurements that we had, shows that it's kind of closer to modern humans than it is to Neanderthals, which again is interesting because genetically, of course, this was not a Neanderthal. It was something completely different. In 2010, Krause and colleagues published a very nice paper that showed that this finger bone from Denisova cave is not a modern human, here in gray, not a Neanderthal, but a completely separate population. Based on the mitochondrial data at the time, we proposed that it's about 1 million, that it's separated from modern humans and Neanderthals about 1 million years ago. Nuclear data showed a slightly different picture. Nuclear data actually showed that Denisova is a sister group to Neanderthals, and we decided to call this population the Denisovans. This is from a slightly later paper from a high coverage Neanderthal genome, also found at Denisova cave, but it kind of shows the relationships between these different populations. The important thing that Denisova really showed us is that the gene flow that we see between Neanderthals and modern humans is not a unique thing. What we saw with in the Denisova genome was that there was gene flow between Denisovans and recent humans, both into the ancestors of Oceanians and into present-day Asians, but that there was also gene flow between Neanderthals and Denisovans, and there was gene flow from a potentially unknown hominin, the so-called super archaic population, into these Denisovans. At the time, we were kind of guessing what this potentially unknown hominin could be, and what we kind of proposed is that maybe this is Asian Homo erectus. But there are also other fossils in Denisova. This here, Denisova 4, is probably still one of my favorite fossils. After the discovery, after the first mitochondrial DNA results in early 2010, Svante Paavo, Johannes Krause, and I traveled to Novosibirsk to talk with our Russian colleagues about the implications of this discovery. And I remember in our meeting, Anatoly Derevyanko just put his finger into his pocket and pulled out this tooth and held it out to me and said, "What do you think of this one?" So this tooth here, Denisova 4, is an absolutely immense upper molar. Still not completely sure whether it's an upper M2 or M3. These days, I'm more likely to think that it's an upper M2, primarily because, as we'll get to later in the talk, we have a couple of mandibles that are likely coming from Denisovans from other sites, and they are all lacking the third molar. And so one of the things seems to be that maybe Denisovans don't have a third molar. This would also explain the very large size. If you only have 2 molars, it makes more sense to have larger M1s and M2s if you don't have an M3. So what is remarkable about this molar is, number one, the size of the occlusal surface, the complexity, lots of additional cusps, and a very massive and flared roots. I'll get to more detail on dental morphology in a moment when I show all the teeth. In 2010, in the summer, we found this tooth here, which at first almost looked like a cave bear. It was broken into pieces, and the first guess of the paleontologist was that it's a cave bear deciduous molar. But in the end, it turned out to be a Denisovan as well, and this was work that Susanna Sawyer, who's now in Vienna, led at the time, and we published it in 2015. Again, very complex occlusal surface. Of course, we can't say anything about the roots, and extremely large size. All of these are in the range far above Neanderthals or practically all late Pleistocene modern humans. We also have a couple of teeth that are not published in detail yet. One of them is this here, Denisova 25, which hopefully will be at least the preprint will be online in a couple of weeks. Another upper second molar As you can see, the roots still massive, but a little bit less flared. Occlusal surface somewhat simpler, and interestingly, very similar to the shape of the molar that we see in the M2 of Harbin, which is a cranium from northern China. Again, I'll mention it later in more detail. That is also a Denisovan. These two look relatively similar. Of course, the strong occlusal wear in Harbin limits our interpretation a bit. And then finally, we also have genetic data from this specimen. So Stéphane Perrin, who did the genetic work on this, let me show you this graph. If you were at the Xiahe, you might have seen his talk about this. So what's interesting about Denisova 25 genetically is we have a complete genome from it, and it's quite a bit earlier than Denisova 3 and Denisova 4, at probably around 200,000 years ago. And also seems to be on a slightly different branch of Denisovans. Another thing that's interesting is we see in Denisova 25 traces of admixture from Neanderthals. Some of the ancestors of Denisova 25 already met Neanderthals probably in the Altai, potentially, and admixed with them, even though this admixture is a couple hundred generations before this individual lived. And Denisova 8 is genetically very close to this individual as well. Denisova 2 is probably basal to all of these. So Denisova 2 is the deciduous molar. So again, we see a very complex population history. Neanderthals and Denisovans seem to keep on meeting, and every time when they meet, they recognize each other as potential mates. Meaning that they kind of saw a shared humanity, which to me I think is the most important result of all these analyses, is that all of these species, or if you want to call them species, all of these populations saw each other as fellow humans, which is very, very cool. We also have one more tooth, Denisova 29, a right upper M2, and I was especially happy when this one was found, because this is actually the antimirror of Denisova 4. It's the opposite side. It's practically the mirror image, a little bit broken, but otherwise practically indistinguishable. And mitochondrial DNA data at least is identical as well in both. So this seems to be the same thing. But you see the massive, especially the lingual root, is always very, very large and strongly flared. And you see the occlusal surface with lots of accessory cusps, etc. So if you compare these four teeth, we can actually, number one, we have the disappointing thing that we only have upper molars for some reason from Denisova. Who knows why? It would be nice to have some lower ones too. But you see that they all share the large size, the complex occlusal surface, and this shape, which is in general relatively rounded or even pentagonal. In size, just to show you a little bit, so this is buccolingual length versus mesiodistal, buccolingual breadth versus mesiodistal length. And if you look in modern humans, in blue here, upper Paleolithic modern humans, in green, early anatomically modern humans, in red, Neanderthals, and then you have Denisova up here, with Denisova 8 being firmly in the range of Australopithecus africanus and afarensis, well above any kind of even Homo habilis, which, in my opinion, is quite remarkable. There's a single Neanderthal that's somewhat higher, and that's Obi-Rahmat 1, which is a weird tooth that probably results from the fusion of the M2 and M3 in the course of development. It's clearly pathological and probably should not be, should be kind of discounted. Then again, I recently had discussions with some people who think that maybe Obi-Rahmat is a Denisovan as well, which would also solve the conundrum. But still, that tooth is clearly pathological. What's interesting in Denisova also is that we see something that we also see in Neanderthals, and that's that the volume of the enamel is relatively low compared to dentine and pulp volume. In general, as you can see, the two Denisovan teeth, so this is Denisova 4 and Denisova 25, 8 is too worn and 29 is too broken to be included here. These two Denisovan teeth are are extremely large. I mean, both the enamel volume and the dentine and pulp volume in Denisova 4 is above what you see in anyone. Same if you look at relative enamel thickness compared to crown volume. Both of these, they have relatively thin enamel. This was always thought to be, or is usually seen as a derived trait of Neanderthals. One of the possible explanations that have been proposed is that because Neanderthals tend to have larger teeth than modern humans, you have the same amount of enamel kind of spread out over a larger tooth. Now, as Denisovans have even larger teeth, this might be an explanation for that as well. One of the interesting things is that somebody looked at a certain, remember the exact gene, that is supposedly linked to enamel thickness and reconstructed enamel thickness in Denisovans as thick. That is probably not true. We also have other material, sadly rather fragmentary. We have this parietal fragment found in 2 pieces. Sadly, the rest of the cranium is missing, which is as usual, kind of heartbreaking. As you can see, we have part of the lambdoid suture. We have a fragment, a piece, clearly the place of a Vormian bone that is missing. We have, of course, part of the sagittal suture, so we can actually estimate the position of lambda. We also have this weird depressed place there, which originally we thought about might be carnivore damage, but on closer examination, we think it's actually from the excavation. Maybe there was a nail in the level above that was put in and went in a little bit too far. Again, these things you don't know, and of course, these two are sampling for DNA. Morphologically, the piece is remarkably primitive. It has a massive temporal line. Actually, we could call it an angular torus. It's so strong here. It indicates that it's a very low, flat, and very wide cranium. It's also extremely large and extremely thick. So especially if you quantify thickness near lambda, it's far above anything you see in Neanderthals. You see even in modern humans, in most Middle Pleistocene specimens, and actually falls into the range of variation that we usually see in Homo erectus. Yeah, it's here you can see it's this is Denisova 13, far above upper Paleolithic Neanderthals. There's one early modern human that comes close, but in general, it's in the range of Homo erectus. You also see that the diploe is responsible for this expansion in thickness. It's not that the lamina externa and interna are thicker, which again is similar to what you see in Homo erectus. So working with Philipp Gunz and Simon Neubauer, we tried to do a virtual reconstruction of the specimen. We used Kabwe, one of the better known Middle Pleistocene specimens, as the template for a reconstruction. Because it seemed to be the most similar. Now, if you know Kabwe, it's a very large cranium. We had to scale up Kabwe by 13% to fit it to Denisova 13. So it was clearly a very large cranium. There are of course many limitations. We only have a small portion. We have to mirror image it. We of course have no idea how the front part of the cranium looks like. We tried 3 different variants for our virtual reconstruction. One of them was kind of the best fit to Kabwe. One was slightly wider and lower, and one was slightly narrower and taller. And then we put a bunch of landmarks. Simon put a bunch of landmarks on the internal surface on the endocranium, and we compared it to a number of fossil hominins. And what was interesting here is that this is a between-group principal component analysis that separates modern humans and upper Paleolithic humans very well from Neanderthals and from Homo erectus. And Denisova 13 plots here not with Neanderthals, but closest to some Homo erectus, in particular to a specimen called Ngawi, which is a very late Homo erectus from Indonesia, which kind of poses the question, whether or not some of these specimens that we have in Southeast Asia that are seen as very late Homo erectus might not be the same population as Denisovans. This is something that Yusuke Kaifu has also discussed in the past. I think that there's some there's some merit to this idea. I do think we have to look in a bit more detail. Hopefully one day we will be able to get molecular data from some of these specimens from Southeast Asia, or even better, found more complete Denisovan remains, of course, ideally in Denisova cave, which you know, we'll see, maybe someday. We also have another parietal, which has been the bane of my existence since it was found, because it's completely useless. So this is the center of a parietal, and some animal chewed or gnawed all around, removing every trace of suture. So you have a kind of roundish piece in the middle of the parietal bone, and you can do fuck all with it. So it's not very useful. Interestingly, it's quite a bit thinner than Denisova 13, and does not show the marked temporal line. So we're not completely sure. This may be because it's a juvenile. We're still working on it. We also have another fragment from Denisova that was found by Tom and Katarina using zooms. And it's a radius midshaft of an about 10 to 12-year-old juvenile, maybe 8 to 12-year-old juvenile, which shows some interesting pathologies in these weird porous. There's like a porous lamina that's deposited on the surface. In recent humans, this can happen in the case of certain vitamin deficiencies, anemia, similar things. We're not 100% sure what's the reason for this. Again, of course, it would be nice to have a bit more, but it's an additional Denisovan individual. So these are the fossils from Denisova. I think what we need to think a little bit about is what we have around. What are other Denisovans? And up until 2019, we did not know any Denisovans from outside the Denisova cave. There were some fossils that some people thought might be Denisovans. I'll show you some later on, but there were no definite Denisovans around. Here I'm showing just a selection of fossils that are either proven to be Denisovans using molecular methods, or are thought to be Denisovans, and also a few of the Central Asian Neanderthals. Of course, in the Altai we have Okladnikova and Chigirskaya caves, both of them with quite a few Neanderthal remains, especially in Chigirskaya. We have about 85 or almost 90 fragments of Neanderthals. We have Teshiktash in Uzbekistan. And then we have two sites in Central Asia, Obi Rahmat and Selungur. Obi Rahmat, when we described it in 2008, we proposed it's a Neanderthal. But as I said, I've been discussing with people recently, maybe it is something different. There's also some archaeological arguments for this. The lithic industry in Obi Rahmat is very different from what we see any Neanderthals making, whether in Central Asia or elsewhere. Selungur is an even more difficult case. Selungur in Kyrgyzstan is a cave site where in the 1980s, excavations by Islamov yielded a bunch of teeth that were described as Homo erectus under the name Ferganthropus. Now these teeth all turned out to be deer and bear teeth. It's very clear that they're deer and bear teeth. But there is a child humerus that is extremely robust, again, coming from a 10 to 12-year-old child. And sadly, we've been unable to get any molecular data from it. Morphologically, all you can tell, it's not a modern human. It's far too robust for that. Extremely thick cortical bone, very strong mediolateral flattening, which is something you see in Neanderthals as well. But the cortical thickness is actually higher than what you see in Neanderthals and comparable to Turkana boy, actually. So we hope that if DNA didn't work, proteins will be helpful. But sadly, the climate in Central Asia sensu stricto seems to be less good for DNA preservation than in the Altai. There are 3 sites where we have molecular data confirming the presence of Denisovans. That's by Xiahe, first cave in Xiahe in China, the Harbin cranium from northern China, and the Penghu mandible from the Strait of Taiwan, and we'll look at those all in a bit more detail. There are also a number of other fossils. There's a tooth from Tam Gu Hao or Cobra Cave in Laos that was published 2 or 3 years ago. where based on morphological comparisons with Penghu and by Xiahe, the authors proposed that it might be a Denisovan. It's in some ways Neanderthal-like, but also in some ways different. So I think that's definitely a possibility. There's a cranium from Narmada or Hathnora in India that also has been proposed repeatedly to be potentially a Denisovan. And the number of fossils from China from Shujiayao, Yinyushan, Dali, Fuchang, and Maba. And we'll look at those in a bit more in a moment. So probably the most spectacular place where Denisovan stayed or lived is Baishir Karst cave in Tibet. This is Xiahe, and I had the chance, was it a year and a half ago or so, to visit it. Absolutely gorgeous at very high altitude, which is very interesting. In Denisova cave itself is at about 750 meters above sea level. But one of the studies that came out soon after we published the Denisovan genomes by Emilia Huerta Sanchez and colleagues was that modern populations living at high altitudes in Tibet carry some Denisovan genetic variants in the EPAS1 gene. And in these populations, this has been selected for strongly and is linked to an adaptation to high altitude. And so there was this question, did Denisovans live at high altitude? And Denisova cave itself was not a high altitude site. And 2015, when this was published, the oldest evidence for human presence on the Tibetan plateau, or anywhere far over 2,000 meters, was after the last glacial maximum. Since then, there's been several sites. One is a site only with the lithic industry dating back to well over 30,000 years. And then the other site is Baishir Karst cave here, which is at over 3,400 meters above sea level. So really, really high altitude. You notice it when you have to climb up these stairs. This is the view from the cave. The cave has been used as kind of a Buddhist sanctuary for hundreds of years. And actually, the fossil Baishir 1 was found by a Buddhist monk meditating in the cave in the 1980s. He found his mandible. He took it with him. And then in around 2016 or so, handed it over to archaeologists at one of the local universities. So this here is the cave. It's lovely. It's full of outside. It's completely full of groundhogs, marmots. They're very cute. They also spread the plague, so don't pet them. I was very happy to see them all around there because one of the interesting things in Denisova cave is actually, if you look at the subsistence in horizons where we have Denisovans, marmots are extremely frequent, often with cut marks. So Denisovans were eating them, and it's also true here in Baishir Karst cave. Marmots were a well-liked portion of their diet. And I was told by a French colleague who grew up in the Pyrenees that actually they cook them too. It's a very tasty stew made of marmots. Whatever. So this here is the mandible that was found in the 1980s. As you can see, it has a very low mandibular body, very robust mandibular corpus, very large teeth. When it was described in 2019, morphologically, they said it shows some Neanderthal-like features, but also features that are much more primitive. But most importantly, they had some molecular data, proteins from the dental enamel that aligned it with Denisova. The teeth also have a few weird features. The lower molars that are usually two-rooted were, in the case of the second molar, actually three-rooted, a so-called radix entomolaris, which is an interesting feature because this is something we see actually today. If you look in recent populations, the highest frequency of this trait is in East Asian populations today. So Shara Bailey and colleagues proposed that maybe this is retention from Denisovans. I'm extremely skeptical of this because present-day East Asian populations carry about 0.1% Denisovan DNA. It's unlikely that the presence of a third mandibular root is one of these, but who knows? But it's interesting because this feature also aligns it with the Penghu mandible from the Strait of Taiwan that we'll see in a moment. So this is the enamel proteins that were described by Fahu Chen and colleagues in 2019. And in this phylogenetic tree, Xiahe is aligned directly with Denisova. and not with Neanderthals, which is, quite neat. Since then, we also have mitochondrial DNA from the sediments from this site that are also Denisova-like. Interestingly, it's actually, most of it is closer to the later Denisovans from Denisova cave, Denisova 3 and Denisova 4, and not to the early Denisovans. The other mandible, as I mentioned, from the Strait of Taiwan, from Penghu, from the Penghu Strait. This is cool because this mandible was actually found by fishermen dredging the bottom of the sea. They pulled up this mandible. So we don't have any kind of real context. The dating is extremely unclear, but when Chen and colleagues in 2015 published it, it was clear that it's an extremely massive mandible. It does not look typically like Homo erectus, but it also is definitely not a modern human. It's something more archaic. And already when it was found, I thought there's a chance that this might be a Denisovan. Here you can actually see the third root in the M2, the radix entomolaris, just like in Xiahe. In general, in all of its morphology, it's a very good fit for the Baixia karst cave specimen. These really belong together. And just a few weeks ago, whenever spring, Xiahe and colleagues published proteins from the enamel of Penghu as well that shows that it aligns with Denisova to the exclusion of Neanderthals. So I think we can be pretty sure here. Probably the most spectacular specimen that is linked to Denisovans is this cranium from Harbin. Again, with a very interesting discovery story. Originally found in the late 1930s while the occupying Japanese were building a bridge there. And one of the workers at the bridge found this cranium while digging and took it and hid it. And it was hidden in, I think, his well, till on his deathbed, he told his grandchildren where this specimen was, and they pulled it out. And finally, it was published in 2021. As you can see, extremely robust supraorbital area, face projecting, but not Neanderthal-like. You don't have the midfacial prognathism. You have a clear malar notch, traits that are kind of more primitive. In general, the whole specimen extremely large. It's the largest fossil hominin cranium we have. It's about 15% larger than Kabwe, which fits very well with our scaled reconstruction of the Denisova parietal. So I was very happy with that. And similarly, you see the very marked temporal line, kind of angular torus-like here, a little less extreme than in Denisova, but going in that direction. It also preserves one molar and M2 here that is sadly very worn, but in its general crown outline, based on the pictures that were published, I was unable to see this specimen up until now. Based on the published pictures, it looks very similar to Denisova 25. It's a pretty decent fit. In the paper where they published this cranium, they also published phylogenetic analysis, which has some interesting results. One of the things that they show is that Harbin is in its own monophyletic group. They group it with Xiahe, with Dali, Yinyushan, Hualongdong, so various middle to late Pleistocene Chinese specimens. But they propose that this group is closer related to modern humans than Neanderthals are, and actually also closely related to Homo antecessor. Now, this is, of course, completely at odds with the genetic data that we have, and I think is not very reliable. They also proposed a species name for this specimen, Homo longi, and at the very end of the talk, I'll talk a little bit about what I think about the taxonomic status of Denisovans and various species names that have been proposed. So in 2021, they described the cranium, and this summer, Xiahe Fu and colleagues published mitochondrial DNA from dental calculus of this specimen, as well as the proteome of dental enamel. And in both of those, Harbin seems to be close to Denisovans. In this case, interestingly, unlike Xiahe, that's closer to the late Denisovans, to Denisova 3 and 4, Harbin is closer to the earlier Denisovan group, Denisova 8, 2, and they also include here 20, 19, and 21. These are all very small fragments that are only known from zooms, and so morphologically not informative. And if you look at the enamel proteome, again, it aligns with Denisova. So I think this is a pretty clear indication that this specimen belongs to the same population as Denisova. But there are a bunch of other specimens in China too that are actually very, very similar to Denisova, and probably for me the closest morphological analog are dental and cranial remains from Xiahe. These are dated to somewhere between 150 and 120,000 years ago in northern China. And probably the nicest specimen is this maxilla of a juvenile here. The M1 is erupted, the M2 is still in the crypt, and a lot of features of these teeth are extremely similar to the Denisova material. So Xing and colleagues published this in 2014. If you look here, this is the M1, this is the M2. If you look at the roots, they are remarkably similar to what you see in Denisova 4, and also the complexity of the crown, the large size, the thin enamel are all features that make this, in my opinion, really the closest morphological analog. Of course, it's also being in northern China, 120 to 150,000 years ago. It's also in the right place and at the right time. This material was proposed to be, again, a different species, Homo juluensis, and we'll get to that in a moment. I'm not going to talk about all the other Chinese material because there really is a ton, but it's very clear that there's a large number of specimens in China that are Denisovan-like. So the taxonomic position of the Denisovans. I'm going to walk through the species that have been proposed for Denisovans, kind of in reverse chronological order. So I'll show the most recently proposed one. So Homo juluensis is the species that was proposed based on a material from Xiahe and another site called Xuchang. This was published in a very weird way because it's in a book by Bei as a sidebar, but the book is by Bei, but in the sidebar they say that this should be cited as Wu and Bei. So Homo juluensis species novum, they proposed that this species the name comes from the Chinese language. It means big head, and we should refer to them as julurens. And they used the Xiahe hominins as the holotype. The diagnosis are a large cranial capacity with a wide and low cranial shape, small and inwardly directed mesoid processes. The most important, the derived Neanderthal pattern of the labyrinth, weak occipital torus, large crown size of teeth with well-developed tuberculum dentale, wide mandibular ramus, and so on, and a prominent temporal line that ends at a distinct bifurcate linear angular torus. All things that we actually see in the Denisova material. So, I'm okay with this. The problem comes in when they kind of describe the hypodyme, the fossils they assign to this, because they also assign to it a bunch of fossils from Denisova that are very clearly Neanderthals, including Denisova 5, the toe bone from which we have the high coverage Neanderthal genome. That kind of is not a great idea. The other problem, of course, with this species is that it, having been proposed in 2024, it's a junior synonym to lots of others. Of course, the other one is Homo longi, based on the Harbin cranium in 2021 by G and colleagues, who give a very detailed description of this cranium and propose why they want to call it longi, which means dragon man. Now, the important thing is, in taxonomy, the older a name is, the higher priority it has. So we have to look at earlier names that have been proposed for this material. And there's actually a name. I found the earliest version of the mitochondrial DNA paper from on the Denisova phalanx that we submitted in 2010. And actually, in this version, we proposed the species name, Homo altaiensis, based on reviewer feedback and various other things. We changed that and just referred to it as the Denisova population, or the Denisovans from there on. But this name stuck with some of our Russian colleagues. And so, for example, Derevianko has used this since then in various publications when he talked about Denisova. The problem is because there is no official species definition, there's no diagnosis, nothing. This is what we call a nomen nudum, so a naked name, a name that is kind of useless, so it should not be used. But of course, sometimes I bite myself why we did not push back against the reviewers and stay with the species name. There are other fossils, though, that could potentially represent Denisovans, and they do have, again, valid species names. For example, the cranium from Narmada in India has been described in 1984 by Sonakia as Homo erectus narmadensis, as a perfectly valid species or subspecies designation. But that means that Homo narmadensis would actually have priority over Altaiensis, Longi, and so on, if we can show that Narmada is indeed a Denisovan. Same is true with Dali. In 1981, Wu proposed a species name, Homo sapiens daliensis. In this case, the problem is that he proposes this conditionally, and that could mean that the name is not actually available. Same is true for the Mappa cranium from southern China that in 1965 Kurt proposed as Homo erectus mappaensis, but again, it's conditional. He says möglicherweise, so potentially in his description, which means that if you take the Code of Zoological Nomenclature seriously, it's probably not available. There's one more name, though, and that's Hemanthropus pei, which is a species name that Königswald, actually a genus and species name, that von Königswald proposed for a number of teeth from Chinese pharmacies. Now, some of these clearly represent orangutans. There's even one that is probably Gigantopithecus in there. But the one that he listed as the holotype, number 7 here, actually looks relatively similar to some of the Denisovan material. And so I've been working with Timon Zanoli and some others on looking at the EDJ of the specimen in comparison with the Denisova specimens. And so if you can show that this specimen is a Denisovan as well, then probably the best name for Denisovans would be Homo pei from Königswald 1957. So if we kind of summarize this, Judulensis is really one that I think we need to forget because Longi was published 3 years earlier, so Judulensis is clearly irrelevant. Altaiensis should not be used, same with Daliensis and Mappaensis, but Homo narmadensis and Homo pei are potentially both valid names in case that the holotype is Denisovan. So I think if we want to have a good scientific name for Denisovans, we kind of need to work ourselves up on this list. Starting with this one, if we can show that this is a Denisovan, good, then that's the name. If it's not, then we have to do the same with Narmada. And then if that doesn't work either, then we're probably stuck with Homo longi, which is Dragon Man is not a bad name either. It's kind of cool. Anyway, so thank you very much for your attention. I want to thank all the people who worked on all of this with me. I mean, without all the genetic work and the help, especially of my Russian colleagues who gave me access to this material when I was a PhD student, which is kind of incredible. This would have been all impossible. I also wanted to just show one historical picture that Katrin brought to my attention recently. This is from 2002 when the Denisovan Oplodnica fossils came to Vienna. And Horst Seidler in the back here organized a press conference with the, at the time, Minister of Science, Elisabeth Gera. And so you see here Anatoly Derevianko, quite a bit younger. Lena Pankeevna, who's his translator, Katrin, of course, and me, 23 years ago, slightly younger. Anyway, so as you can see, Denisova has a very old history in Vienna. And so I'm very happy to be back here and talk a bit about this. Thank you.